Have you ever just looked at, I don't know, a giant redwood tree, or even just your own hand and wondered, how did all of this come from a single tiny cell? Well, the secret is locked away inside a molecule. A molecule so powerful it acts as the master blueprint for every living thing. It's literally the software that runs the hardware of life. And today, we are going to unravel its code piece by piece.
So here's how we're going to break this down. First, we're going to jump in a time machine and go back to the hunt for the secret of life itself. Then we'll put that secret under a microscope to really see its elegant double helix structure. After that, we'll zoom out to appreciate the absolutely incredible challenge of packaging that blueprint. And finally, we'll discover the epigenetic switches that act as the director, deciding which parts of the blueprint actually get used. Sound good? Let's go.
All right, our story kicks off in the early 20th century, and you gotta understand this was a time of intense scientific mystery. Scientists knew that traits were passed down from parents to children, but they had no clue what was actually carrying that information. Was it a protein? Maybe RNA? The hunt for the molecule of heredity was officially on. As you can see, this breakthrough did not happen overnight. It all started back in the 1920s with Griffith's experiment. He saw that some kind of mysterious transforming agent could turn harmless bacteria into a deadly version. Now, for years, the number one suspect was protein. But then, in the 40s, Avery, McLeod, and McCarty did some serious detective work. They proved, methodically, that this agent was in fact DNA. The final crucial piece of the puzzle came in the 50s, thanks to Rosalind Franklin's absolutely incredible X-ray image. It gave Watson and Crick the clue they needed to build their 1953 model, the iconic double helix we all know today.
You know, I just love that term they used back then, the transforming agent. It sounds like something out of science fiction, right? But it perfectly captures the mystery they were facing. They could see the result, harmless bacteria suddenly becoming lethal, but they couldn't see the cause. It was like a ghost in the machine, and the race was on to figure out what that ghost was made of.
So just think about the huge shift in thinking this was. For decades, pretty much everyone assumed the blueprint of life had to be some super complex protein. But the evidence was undeniable. The secret wasn't held in a complicated molecule with 20 different building blocks, but in DNA, a molecule with just 4. I mean, it's kind of counterintuitive. This discovery basically launched the entire field of molecular biology, and of course brings us to our next big question. What does this incredible molecule actually look like up close? Okay, so now that we know DNA is the star of the show, let's zoom in and get a really good look at its structure. Because it's not just some random string of chemicals, not at all. It is a masterpiece of information architecture, brilliantly designed for stability, for replication, and for storage. So it all starts with this one fundamental unit, the nucleotide. The easiest way to think of it is like a single Lego brick. It has 3 distinct parts. You've got a sugar molecule called deoxyribose. That's where DNA gets the D in its name. Then attached to one side of that sugar is a phosphate group. That's going to be super important for linking all these bricks together. And on the other side is the real business end, the part that stores all the information, one of 4 nitrogenous bases. So how do these individual Lego bricks build a long chain? Well, it's kind of like building a freight train. The phosphate group of a new nucleotide, our new train car, hooks up to a very specific spot on the car in front of it. This creates this incredibly strong connection called a phosphodiester bond. Now, because of the way they connect, the chain can only be built in one direction. It has a definite front, which we call the 5' end, and a definite back, the 3' end, which is where new cars are always added. This one-way street, this 5' to 3' directionality, it is absolutely critical for how our cells read and copy all those genetic instructions. Okay, now let's talk about those nitrogenous bases, the actual letters of the genetic code. They come in 2 families. You've got the purines, that's adenine and guanine, and they have this double ring structure, which makes them a little bit bigger. Then you have the pyrimidines, cytosine and thymine, which have a single ring, making them smaller. And this difference in size, it is the key to how they pair up. And right here, this is the central rule of DNA structure. It's beautiful in its simplicity. To keep the latter a perfectly uniform width, a big purine always has to pair up with a small pyrimidine across the middle. But the rule is even more specific. Adenine only ever pairs with thymine, and they're held together by 2 hydrogen bonds, and guanine only ever pairs with cytosine, and they use 3 hydrogen bonds, which are a little stronger. This perfect complementary pairing is the secret sauce to DNA replication. If you have one strand, you automatically know how to build its perfect mirror image.
So let's put it all together real quick. What do we have? We've got two strands that run in opposite directions. We call that anti-parallel. We've got a strong sugar phosphate backbone that forms the outside rails of our twisted ladder. In the middle, you have those complementary bases, A with T, G with C, forming the rungs. And as the whole thing twists into that famous helix, it creates these grooves, a wide major groove and a narrower minor groove. Now, these grooves aren't just an accident of the shape. They are essential docking bays where proteins can attach to read the genetic code inside.
Okay, so we've seen the elegant design of the blueprint. Now for what might be the real mind-bending challenge. How on earth do you fit an enormous, sprawling instruction manual into a space that is smaller than a speck of dust? This is a molecular engineering puzzle on a scale that is almost impossible to wrap your head around.
Seriously, just try to picture this for a second. If you could take all of the DNA from one single human cell, a cell so small you can't even see it, and you stretched it out from end to end, it would be about 2 meters long. That's over 6 feet, taller than most people. An absolutely immense library of information tucked inside every one of your cells.
And where does that 2-meter-long string have to go? Into the cell's nucleus, which is only about 6 micrometers in diameter. To put that into perspective, that is like trying to stuff 25 miles of incredibly thin thread into a tennis ball. I mean, how is that even possible?
Well, the cell's brilliant solution is a substance called chromatin. Your DNA doesn't just float around in the nucleus like a tangled mess of spaghetti. No way. Instead, it is meticulously, carefully wrapped around a scaffold of proteins. And this combination of DNA and protein, that's what we call chromatin. It is the very stuff our chromosomes are made of. This incredible compaction happens in stages, as you can see here. First, the basic DNA double helix gets wrapped around proteins, creating what looks like beads on a string. But that's just the beginning. That string of beads then gets coiled up into a much thicker fiber. Then that fiber is organized into these massive loops, which are coiled and condensed again and again and again until you finally get to the most compact form, that classic X-shaped chromosome you see right before cell divides. Every single step is a true marvel of engineering. Let's zoom back in on that very first level of packaging, the beads on a string. Each one of those beads is a structure called a nucleosome. This is the fundamental, basic unit of chromatin. It's made of a very specific length of DNA, about 147 base pairs, wrapped almost twice around a central core made of 8 special proteins called histones. Now, this histone core is a perfectly organized little protein machine. It's an octamer, which just means it's a bundle of 8 proteins. You get 2 copies each of 4 different types: H2A, H2B, H3, and H4. Together, they form the perfect little spool for the DNA thread to wind itself around. Then, a 5th histone, H1, comes along and acts like a clip. It fastens the thread to the spool, locking it in place and helping to bunch all those spools tightly together, getting them ready for the next level of compaction. And now, for what might be the most dynamic and exciting part of our whole story. This unbelievably intricate packaging isn't just for storage. It's also a highly sophisticated control system. The cell can actually tighten or loosen different sections of its chromatin to control which genes are read and which are silenced. This is the amazing world of epigenetics. So basically, your chromatin exists in 2 main states. On one hand, you have euchromatin. Think of this as the active library. The DNA is loosely packed, it's accessible, and the genes are open for business, ready to be read. On the other hand, you have heterochromatin. This is more like the deep archives. The DNA is packed so tightly that the genes are completely silenced, locked away in storage. The amazing part is that the cell can switch between these states to control which books are being read at any given time. So how does the cell decide which parts are open and which parts are closed? It uses something called the epigenetic code. And this is a totally fascinating layer of information that's written on top of the genetic code. So instead of changing the A's, T's, C's, and G's, the cell adds or removes these little chemical tags to the DNA itself, or more often, to those histone proteins the DNA is wrapped around. These tags act like tiny sticky notes, telling the cell's machinery, hey, read here, or nope, ignore this part. And this code is incredibly specific. Those histone proteins have these long tails that stick out, and these tails are like little command panels. As you can see in the table, adding one type of chemical tag in a acetyl group can loosen up the DNA and turn a gene on. But adding a different tag, a methyl group, to a different spot can do the complete opposite, tightening the DNA and turning the gene off.
By reading these different combinations of marks, the cell can fine-tune its activity with just stunning precision. And this isn't a set-it-and-forget-it system, not at all. It is constantly changing. Biologists have this great analogy for it. You have writer enzymes that add these epigenetic marks, you have eraser enzymes that remove them, and you have reader enzymes that recognize the marks and then tell other proteins to either activate or silence the genes in that area. It's a living, breathing, dynamic conversation happening on your chromosomes at all times. And this all leaves us with a really fascinating final thought to chew on. Our DNA blueprint, the sequence of A's, T's, C's, and G's, is essentially fixed from birth. But the way it's interpreted is flexible. It's constantly being rewritten by our lifestyle, our environment, even our experiences. So if the DNA is the hardware and epigenetics is the software, it raises a pretty profound question. How much of you is written in permanent ink, and how much is a story that you are still writing right now?